Welcome to the Escape Room Prison Cell, a prison room you escape with knowledge about cells. Contestants play in groups. With each question you answer correctly, you will be closer to reaching the exit. There will be 3 rounds of questions, each round with slightly different rules. Let's begin the game. For the first round, a speaker will be selected at random. All players can discuss the answer, but only the speaker will be able to submit the final answer.
Plasma is quite impatient, so it better not be him. I don't want to do the work, so it better not be me.
The speaker chosen for this game is...
Not plasma.
Plasma. First question. What are the 3 components of cell theory?
Oh, that's easy. I don't understand the question. Cell theory is a set of basic principles of cells. It is made of 3 principles or ideas. Cells are the basic units of life.
Cells are the basic units of life.
All cells arise from pre-existing cells.
All cells arise from pre-existing cells.
And all living things are made of 1 or more cells.
And all living things are made of 1 or more cells.
That's it.
That's it.
Your answer was recorded. That's right.
A related concept is that all organisms can perform basic life functions, such as growth and metabolism.
Even organisms that are made of 1 cell.
Yep, Paramecium and Chlamydomonas are good examples of unicellular organisms that can carry out life processes.
Second question. One of these images is from a light microscope and one is from an electron microscope. Which one is which?
How are we supposed to know that?
Electron microscopes use electrons to produce an image, while light microscopes use visible light. Electrons have smaller wavelengths than visible light, so they can help us see smaller things.
How small?
As small as organelles, like the nucleus or mitochondria.
Oh, so it's this one.
Correct. Next question. Give the magnification of this image.
To calculate magnification, we need to divide the image length of the scale bar to the actual length according to the image. The image length is... Hold on, do we not get a ruler? How are we supposed to know the image length then?
Oh, that is exactly as wide as me. And I know I'm exactly 1 meter wide.
How do you know your width?
I measured myself last week. I grew.
Okay. So the image length is 1 meter, and the actual length is 0.4 micrometers. 1 meter is 10 to the power of 6 micrometers, so we need to multiply the meter by 10 to the power of 6. This leaves us with a magnification of
2,500,000.
That's right. Next question. State a development in electron microscopy that has improved the study of cell infrastructure.
Huh?
One example is cryogenic electron microscopy. This involves freezing a solution of a particular protein, which allows us to see the same protein from different angles. Using this, we get a 3D image of the protein.
Great, so too difficult to pronounce. Are there any other ones?
Yeah, there is immunofluorescence, which is when you attach fluorescent tags to specific molecules, so you can track their location easily.
The name's too long. Any other ones?
There is also freeze factor, which is when you freeze the cell and then break it to get a 3D image of the inside of the cell.
Oh, I like that name. What was it again?
Freeze factor.
Freeze factor.
That is correct. You've now reached the second round. Here, each question is going to be split into 2 parts. In the first part, you will get the topic the question is about. During this time, both of you are allowed to communicate with each other. In the second part, you will get the actual question. During this time, only the speaker is allowed to speak. Any questions?
What's the topic?
It's eukaryotic and prokaryotic cells. You may begin discussing. You have 1 minute.
Alright, Plasma, listen carefully. There are 2 different types of cells. They both have a cytoplasm, a cell membrane, ribosomes, and DNA. Prokaryotes don't have membrane-bound organelles such as nuclei, so all the components are free in the cytoplasm.
So disorganized like me, plasma. P for prokaryotes and P for plasma. Got it!
Yeah. Some other features they may have are a cell wall, cilia, and flagella. Also, prokaryotes have 70S ribosomes.
Didn't you say eukaryotes also have ribosomes?
Yes, but the ribosomes in eukaryotes are 80S ribosomes. Another difference is that eukaryotes do have membrane-bound organelles, such as nuclei and mitochondria.
Time's up. Now, you may not communicate with each other and only the speaker can speak. Here's the question. Which of these is a prokaryotic cell?
Gosh, that's hard. Prokaryote starts with P and P is for plasma. So the prokaryote should be the cell that is not organized into compartments, which is this one.
That's right.
Phew.
The next question will follow the same format and the topic is animal, plant and fungal cells. This time you will only have 5 seconds to discuss the topic. So hurry up.
Plant cells have a cell wall, chloroplast, and vacuole. Animal cells don't.
Time's up. The question is this an animal cell, plant cell, or fungal cell?
It has a cell wall and a vacuole, so it's a plant. But it doesn't have chloroplasts. H2O didn't have time to tell me about fungal cells. It's a fungal cell then?
Correct.
Well done, Plasma.
You have now reached the final round. In this round, you will be separated into 2 rooms, where you will not be able to communicate with each other at all. Both of you must answer the question correctly to pass the round. The topic is the same: animal, fungi, and plant cells. The question is, which of these are fungal cells?
Oh no, this is a hard question. They are all atypical cells. The first picture shows red blood cells which don't have nuclei. They are animal cells. The second picture shows skeletal muscle fibers, which are the cells that make up the muscles around your body.
Are these even cells?
They are really large cells with multiple nuclei. These are also animal cells. The next picture shows a phloem sieve tube element, which are the elements that make up the phloem tube in plants.
When in doubt, always choose C, so I'll choose the third one.
The last image shows cells with cell walls and multiple nuclei. This looks like aseptate fungal hyphae, which are fungal cells.
Both of you have submitted your answers.
No, I misclicked. I didn't click C.
This last question determines whether you win this game. And the result is that both of you got it right. You won the game.
Yay!